DNA replication. How does a molecule of DNA produce a copy of itself? And we want to look at this from a biochemical standpoint. All right. Much of what we're going to learn about DNA replication also applies to RNA replication. It's just that RNA is a single strand versus the double-stranded DNA. So there's some small modifications, slightly different enzymes involved, but we're going to just look at DNA as sort of our main example. So first of all, what I'm going to do is I'm going to draw a little section of a DNA molecule. I'm going to put a phosphate here. Okay, put a P inside, phosphate. And then, of course, that's attached to this carbon, which is attached to the ribose sugar, like this. And the ribose sugar is attached to, I'll just put a circle and we'll call it, say, guanine. Okay? If I was drawing this upside down on the other side, we would have sort of a hydrogen bonding here and a cytosine molecule like this, which would be attached to sort of an upside down ribose, something like this. And then the ribose would then be attached to a phosphate through the carbon over here on the other side of the molecule. So what we get is this directionality of the DNA molecule. The strands are opposites of each other, but they're also sort of running in the opposite direction, right? And so the first thing we have to understand is how we determine the directions of the DNA strands. And it's pretty simple. If you look at the carbon numbers in the ribose, typically this is called carbon 1, 2, 3, 4, 5. And over here, if you do it upside down, right? 1, 2, 3, 4, 5. So the carbon that's closest to this end, if you imagine this as a continuing chain, right, down, going down, guanine, another one with adenine, another one with cytosine, the carbon at this end is carbon 5. So we call this the 5' end, the 5 end. We put a little prime to show it's the 5th carbon that's sort of on the edge of this end. Over here, if you look at this, the carbon is right here. This is the one sort of on the edge. So this is the edge we call the 3 end. 3' is the little symbol that we use for that. So a DNA molecule has a 5 end and a 3 end, depending on which carbon is sort of closest to that end in the ribose molecule at the end. So that's what we mean by 3 and 5. It's just a way of organizing the 3 end versus the 5 end. Okay? So typically, if you were going this way on this chain, you'd be going in the 5 to 3 direction, right? But if you were running along this chain this way, you'd have to go the opposite way to go 5 to 3. If you were actually running the same way, along with, if you wanted to go in this direction, we'd say that's the 3 to 5 direction. Okay? And that's really just a little bit of geography that we need in order to figure out where we're going to go. All right, now we're going to draw strands of DNA. And to keep it simple, we're not going to draw molecular or structural pictures. We're just going to draw diagrammatic representations. Okay, so what I'm going to do is I'm going to take a color here, 2 colors. I'll use a purple color, and I'm going to draw a strand of DNA that is being partially unwound. Okay, so here's the unwound part. And then what I'll do is I'll just go down like this and wiggle it up and down like that as it coils and twists. Then the other part I'll draw in a green color down below, and then I'll bring it up here like this, And let's bring it up like this and wiggle it up and down as well. So there's your double helix. Okay. And then what I can do is just show the little lines inside. Whoops, I wanted black. These are the hydrogen bonds between the bases, C's and G's, and so on. So this is sort of what the DNA, it looks like a ladder made of rubber that's been twisted, right? Okay. So the first thing, of course, that has to happen if DNA is going to replicate is it has to unwind. It has to sort of produce this unwinded section. And so there's a actual enzyme, which is an unwinding enzyme, and it's called helicase. So what I'm going to do is I'm going to draw a picture of helicase here, sort of like as a blobby red thing around here, and I'll shade it in with a crayon. So there's my helicase. It's wrapped around the DNA molecule. And as it moves along the molecule, it is opening up the strands. So it's obviously moving along this towards our right. Okay, so that's the unraveling molecule. Let's name it helicase. And its job is to unwind the molecule. So helicase unwinds DNA strands. Okay. Now the reason I drew the strands in two different colors is because these two strands, as we saw, are running in different directions. So we're going to put the ends on them. So my top one, my purple one, let me go back to purple here with a small line. My top one is going to have the 3 end here. And if we follow it all the way to the other end down here, this would be the 5 end. I'll just squeeze it in there on the right-hand side. And then my bottom strand would have run the opposite way because it's upside down. So this would be the 5 end. And then over way down here would be the 3 end. So it's important for us to keep that in mind because these enzymes, they actually physically travel along the molecule, and the directions that they go are kind of important. Okay, so now that the DNA is unwound, you can imagine that inside here, I'll just use little bars. What we have in here are the nitrogenous bases, and they're like this. So these sticks stands for the C's and G's, but they've been pulled apart. The hydrogen bonds that were attracting them before have been pulled apart by the enzyme that catalyzes this unwinding reaction, helicase. Okay, so the next step is that we have to somehow start building a new DNA. But we have to build, each copy is going to get, the purple 3 to 5 strand is going to get copied, and the green 5 to 3 strand is going to get copied. And so the first thing that has to happen is that there's another enzyme that's going to come along that's going to do the assembly. But this enzyme needs a starting point. It sort of needs something to get it started. So what first happens is this primase molecule. RNA primers are built by something called primase. So I'm going to put a blue molecule right here, blue enzyme here, and another blue enzyme sort of down here. Now the interesting thing about these enzymes is they all travel 5 to 3. For some reason, the enzymes can only travel 5 to 3 on the molecule. They can't travel 3 to 5. Okay? Well, actually, that's not true. Okay, let me restate that. These ones can travel. They travel 5 to 3, but they're named based on what they're assembling, not what they're riding on. That's the trick. So what happens is this blue guy here, let me color them in. Let me color them in blue crayon, just so we have some sort of depth to them. Okay, and we'll call it primase because that's its name. So we'll go back here, pen, blue, color, small. So this is called primase, this enzyme, and so is this one, primase. And what they are doing is they are assembling or building as they go RNA segments. They're attaching to the previously together DNA molecule. So what's happening in here, I'm going to use another color. Perhaps I'll use green along the bottom of this one. Let's see. No, not green. I want a different color. How about a nice limey green? That's a good color. So this guy has produced a piece of DNA, or RNA, I guess you could say, and this one is producing it here. And these are being sort of attached. They're being made. They're not actually attached yet, believe it or not. They're just being made. And what those are RNA primers. They're little bits of RNA, and they're very short, right? And of course, if you look at this one, this particular one here at the top, this is the 5 end of it, right? Because it's opposite the 3 end of the DNA, and this is the 3 end of it. So this blue primase is traveling in this direction along the molecule. And this one, of course, is traveling in this direction along the molecule, and it's making a little piece here, and this is the 3 end and the 5. So you can see that it's going 5 to 3. It's building 5 to 3 is the way we should say it, I guess. It's not writing 5 to 3, it's building, assembling 5 to 3 as it goes this way. Now this primase only adds a small little piece of RNA. So we're going to write underneath here, primase inserts a small segment of RNA, right? RNA primer. That's what it's called. It's called an RNA primer. And that serves as a starting point for the actual DNA building. For another enzyme, which is called DNA polymerase 3. So, starting point for DNA polymerase 3 to begin assembling the DNA. Sure I can, yeah. We can go slow on this because it's going to, right there. Primase inserts a small segment of RNA primer that serves as a starting point for DNA polymerase 3 to begin assembling the new DNA strand. Why don't we say assembling the new strand? So what you'll notice right off the bat is that a piece of DNA is not just a string of genes. It has a whole bunch of segments and pieces in it that are part of the process of assembling, telling the gene when to code, when not to code, when to turn on, when not to turn on. And we'll talk more about that later. Only some of the DNA is actually genes. There's a lot of other directional, instructional information about how it replicates and turns into protein and so on. Okay, so that's 2 steps so far. The helicase, of course, is moving this way. And that determines the overall direction of the replication process. Right? And so you can see what will happen a little bit later on. If these move a little bit, right? Once these primers are sort of set up, then what has to happen is the DNA is going to be built. And you can see that on the top half, if I put my little primer in here, I can now assemble towards the right and follow the helicase as it unwinds and just keep on going. But on the bottom, you cannot move in that direction. You have to assemble in the 5 to 3, or sort of, you have to assemble in the 5 to 3 direction, which means you have to move towards the right. In other words, on this picture, all the enzymes have to move towards, no, let me rephrase that. You have to move towards the right on the top strand, but towards the left on the bottom strand, the 3 to 5 direction, right? The green strand would have to move towards the left. So that means that on the bottom strand, the DNA polymerase that's going to build the DNA is going to have to move. I'm going to draw a line for those who can't see what I'm doing. It's going to have to move this way, away from the helicase. So that's the key. It's moving in the opposite direction of the helicase as opposed to the one on top, which is following it along. So I just wanted to stress that now because that's going to come up later when we see how it actually has to work. Okay. So now let's draw the same picture or a similar picture, but we're going to draw those enzymes as if they've moved a little further on down the line. Okay? All right. So we're going to draw our top one, which was purple. Okay. So I just got to remember that purple is the 3 to 5 strand. Okay. So let's draw the purple one. So I'm going to have a longer piece of it unwound and just a little bit here because the helicase has moved further down, right? And then here's my green. There, something like that. And we can put in our little DNA markers like that. And then, of course, what we have here are all the DNA bases that were separated. They should be lining up perfectly, but just for kicks, we'll do it like that. I might have missed one or two there somewhere. Okay, so now the helicase has moved further down. So I'm going to draw the helicase over here and color him in a nice red color. Right, he's moved further down. And if you go back to our picture, on the top strand, you'll see that there's a piece of RNA primer right at the start. So I'm going to draw that little piece of primer in on the far left, on the top. That blue primase is inserted it there. So here's the little piece of primer that's lime green. I think it was this color. A little piece of RNA primer right here that was inserted. I need some black. Like this. And if we go up again, we can see that a little further to the right, the other primase on the bottom has inserted a little piece of primer. So I'm going to try to draw that sort of right about here. Right about here. Oops, and that should have been the lime green color, eh? So let me change this to lime green. So those are my two little pieces of RNA primer that the blue enzymes had put in here. Okay? And we should label, this was it 5 to 3, the purple, or was it 3 to 5? Let's double check. I think it was 3 to 5. Yeah, 3 to 5. So let's put the 3 end here on my purple chain and the 5 end here. And then on my green chain, we'll put the 5 end here and the 3 end down here. That means this little piece of RNA in this little green color is 5 end, 3 end, but the one on the bottom is 3 end, 5 end, like that, right? Because remember, they run opposite to the ones they're joined to. Okay. Now on the top segment, a new enzyme comes along. This time I'm going to use, I used blue. Maybe I'll find a nice turquoisey color. This will work well. So once this little RNA is here, this new enzyme can form right here, can join up here. This is the DNA polymerase 3. And I'll shade him in with my crayon, DNA polymerase 3. He travels 3 to 5. He assembles 5 to 3. So what he begins doing is he begins assembling a new piece of DNA from the starting point of that RNA primer piece. And he moves in this direction. Let me just get... he moves this way. So he's following the motion of the helicase, right? And so he has no trouble. He just assembles DNA. And so he's going to move along there and he's going to just put in pieces of base and sugar and phosphate units and complete the strand along the top. The problem is that there's another one that's going to join up on the bottom, if I can do this without too much interference. The other one is going to be down here, like this. And he's going to assemble as well, but he has to assemble the other way. He has to go this way, which is a little bit weird. Get my crayon and the right color, color him in. Meanwhile, what's going to happen up ahead here is that primer enzyme is going to come back and insert another piece of primer right here. So I need the blue enzyme to come back. And he's attaching right here again. Let's color him in. Oh, I've just lost. Oh, here we go. Crayon, color, blue. Let's color him in blue. So he's that RNA primer piece. And he's also going to run along, oh crap, he's going to run along there in that direction. Everything has to go in the same direction, right? So he's going to go along and put in a new piece of primer. So now I'll get the ugly green. Good. And he's now assembling a new piece of RNA primer in here. And of course, he's moving along this way as well. So there's a space between the first primer and the new piece of primer. So what has to happen is the DNA polymerase 3 has to go like this along here, but then another one has to jump in here and work backwards. And so you always need to have little pieces of RNA in between so that another one of these polymerases right here, right, would then have to jump into this place and travel this way up to the next piece. Now, it's a very short piece there that I've drawn, but that's the idea. So what we'll do is we'll show what it looks like when we have a whole bunch of things going on at once. Ah, colors, come on. This is the DNA polymerase there. So while the top piece is running, just this one polymerase can just chug along forever and carry on until it's all over. On the bottom, you have to have a piece going back, and then another RNA piece and a piece that comes back, and an RNA piece and a piece that comes back, you see? So you're only assembling small sections at a time on the bottom. You can't just run the polymerase all the way across. And so this section, right, the section of DNA that's going to be built here is called an Okazaki fragment. Okazaki fragment. So now what I'll do is draw another picture, and I'll draw it smaller and show you what it looks like sort of after it's run for a while. But I'll give you a chance to catch up to me. Okay, so before we draw that picture, we have terms that we use to describe the two strands. We call the top strand the leading strand because it's running in the same direction and it's just doing its thing. And the bottom strand, we call it the lagging strand because it seems to be sort of like a run up and work backwards, run up and work backwards type of approach. Okay, so now let's draw the whole thing in a bit of a smaller format. So purple DNA, and just a couple of spins at the other side, and green DNA. Actually, I just realized if I make this a little bit bigger, it'll make more sense. So let me just kind of bring this down and wiggle it up like that. And then back to my green, right across there, and then wiggle it up like that. So there's the helicase still chugging along, doing its thing. Right? He's still moving this way. Oh, play on. He's still moving to the right, doing his thing. He's still chugging along this way. Helicase, unwinding the protein. Shortly in behind him is the RNA, or sorry, the DNA polymerase 3, which was this weird turquoise color, right? And he's sort of chugging along behind. Let's put him sort of right here, and let's color him in with the crayon. Wow, my smartboard. Not so smart today. Color, turquoise, color, shade him in. Okay. And then what he's created is a whole new strand. So we'll have the original little piece of DNA that was formed way back here, a little RNA actually. It's an RNA piece. And then the rest of this is going to be basically just the green DNA. And it's been assembled by that guy. He's gone all that way and assembled that all the way along. Now let's put in some of our little lines here. So he's laying up these pieces in line. Okay. Now I know those two greens are very similar, but it's important to realize that this little light limey green one is different from the other green. Okay. In fact, maybe what I should do is use an even limier green to distinguish that. Let's try this. See if that distinguishes it a little bit more, a little bit. So that's what's happened on the top strand, right? If we go back to our original picture, you can see the limey green piece that we originally put in, and the primase makes the limey green piece, and then it's done. And then the DNA polymerase 3. So we didn't label him, did we? The sort of turquoisey cyan color. Let's just go back here and just label him. This guy's name right here is DNA polymerase 3. There's different kinds of DNA polymerase, as we're going to see, so it's important to remember that he's 3. Okay, and so on the bottom, we have all these little Okazaki fragments built up. So what eventually happens is we get these limey green sections that were made. We'll put one here, and we'll put another one here. We'll put another one maybe right here. Maybe up there, we still have a primase molecule that is still doing its job, inserting that. So we'll put a blue primase here, showing that he's doing his thing. We'll color him in with a crayon. There's the primase reassembling that little RNA piece. Okay, so let's put the RNA in there like this. And then we can put some pieces along here. Oh, we should have them all the way. These ones are already joined, so let me just rub out the middle a bit. Make those a little smaller. Okay, that's looking better. And then this is already put in. Okay. And what's happened is that the blue polymerases now, I'm going to draw them sort of right at the end of their little session. So give me this turquoise color here. So this one has bumped into this other Okazaki fragment, or this this other RNA primase, this other RNA primer that was put there, right? So he's kind of done. And what he's done is he's assembled behind him this whole piece of the DNA molecule. And he's done it by moving in the opposite direction. He's moving towards this way, right? And this guy's moving this way. Okay, so far so good. Then we have the other, another polymerase, which is hooked up here. Okay, I'm going to make this a thicker line because we're just for our theme here. The other one has moved down to here, off of our picture. He's kind of gone. So let's put him right here on the end. What would happen is there'd be another RNA piece of primer here somewhere because the pattern would repeat. We just didn't have it on our picture. So he's finished making this whole strand here. Okay. So in a sense, you can see all the pieces getting in place, and he was moving this way. He's pretty much done, so likely for him, he's going to detach, and that's the end of his job. Another one will have just begun to build another Okazaki segment behind this primer on the bottom. So we need another one. So it's going to have set itself up here, and it's going to start moving this way, and it's going to just be starting to build its DNA piece along here. And now let's just fill in all the little things that we need to here. Little base numbers. Good. And we'll color in our guys. Color these guys in. Okay. And remember, these are DNA polymerase 3s. So this last one that I've drawn, the one on the right side on the bottom, he's going to continue until he hits this RNA primer that's coming up, right? I can't illustrate that. Maybe if I use the pointer. No, I can't. I don't have a pointer. So essentially, I'll just use a color and erase it. This guy is going to move this way until he stops right here at the last piece of primer. And so he'll fill that in as well. So the end result is you've got a top piece that looks perfect and a bottom piece that is all messed up. So what we'll do is we'll stop this video because we're out of time, and we'll continue the process with how do we fix this bottom piece which looks all chopped up with bits of RNA in it, okay?